The title of the paper we're going to be talking about today is the crystal structure of the dihydroorotate dehydrogenase from Helicobacter pylori with bound riboflavin mononucleotide. And so I'm here with the authors, Ashna Agarwal and John Georgiades. And so we're just going to do some quick introductions. So just say your name and what institution you came from and when you graduated from Washington University. So Ashna.
Yeah. Hello, everyone. My name is Ashna Agarwal. I graduated from Washington University in St. Louis in May 2024. And I was a co-author on this paper. I was responsible for conducting a software-based structural analysis of the crystallized structure of dihydroorotate dehydrogenase from Helicobacter pylori with bound FMN.
Okay, great. John?
Hi, everyone. My name is John Georgiades. I also graduated from Washington University in St. Louis in May of 2024. And I also sort of contributed to the structural characterization of the Helicobacter pylori dihydroorotate dehydrogenase, focusing primarily on analyzing some active site features, as well as one of the key highlights of our paper, trying to understand this sort of channel that we propose ubiquinone traffics through to find the active site.
Right, great. So can you guys tell me, or so Ashna here, can you tell me about your backgrounds and what led you to choose the course that I teach, the structural bioinformatics of proteins, the Bio 4525 course at Washington University? What was your background? What made you, what sort of got you interested in choosing this course over the courses that you could have taken instead?
Yeah, I would love to. Generally, I was a major in genomics and computational biology. Throughout my time at college, I was very interested in learning more bioinformatic tools and generally more computational tools furthering study of molecular biology, protein studying, etc. I think the class that I took under Mr. Smith was a pivotal class in achieving this goal. I'd taken other classes before that began to understand more machine tools and computational tools of understanding biology. But Professor Smith's class was the first time that I really got to investigate, learn, and really conduct my own analysis.
Okay. John?
Yeah, so I came into this course sort of just after starting research in undergraduate research looking at sort of structure-based drug design. And so I came into this course and really chose this course because I wanted a much more robust sort of I guess, background and set of tools to use to approach my own research project. And throughout this course and working on this project in particular, I actually started to sort of develop a greater interest in more specifically enzymology, mechanistic enzymology. I guess that's what happens when you stare at the structure of the same enzyme for a long time over the course of the semester. But sort of from that, I gained an interest in enzymology, and that's sort of the area that I'm now able to do my PhD in.
Cool. So once the protein was assigned to you, or your first steps that you looked into to try to understand it? And well, does anyone does it? I will ask John that question, and then you can add if anything you want to, Ashna.
Sure. Yeah. So I think one of the first key steps once we, sort of knew that this was a dihydroorotate dehydrogenase is starting to look at the literature and what other people have done to characterize this class of enzymes in other organisms, trying to understand the various sort of modalities that these enzymes come in, two major classes. And sort of from that, we began a sort of larger scale comparative analysis, comparing the sequences of these enzymes as well as their structures, of course, since we have this beautiful crystal structure. And so that sort of led us to begin trying to understand some of these relationships between the Helicobacter pylori enzyme and the other dihydroorotate dehydrogenases across biology. And I'll sort of push it off to Ashna to sort of finish out, I guess, that description.
Yeah, perfect. I mean, I definitely agree. The first thing we started with is conducting an investigation into past literature to further understand just the chemistry of the dihydroorotate dehydrogenase proteins. we also continued a visual analysis of our structure the entire time to maintain this correlation between what we were reading and what we were observing. Following our structural analysis across homologs, we conducted a visual analysis as well as prediction analysis to identify if there's any channel or to understand if there's any mechanism by which we see in association with the ubiquinone pool that we had identified in previous literature. So we really tried to understand what the mechanism of entry of ubiquinone would be to the active site of this protein. We used visual analysis, we used PyMol analysis and past literature to support this, and we identified a electrostatic and terminal microdomain that was also corroborated by other DHODH proteins in other microorganisms.
Okay. So you had mentioned this, well, you mentioned the microdomain, but I actually want to sort of talk about this. So your paper, one of the cool things about your paper, I thought was this, so you guys identified this hydrophobic channel that would help facilitate ubiquinone access. So how did you come to that insight, and what role did structure visualization, what sort of tools and things and that you used to help and aid in that discovery? Uh,
Yeah,
but
I can start and then we can have John go into the further characterization that we did. But initially, we had read from previous papers, and we had a general understanding that class 2 DHODH proteins usually use a ubiquinone that was present in the inner membranes. And we realized that the active site pretty early on was in the core of the protein. So we really tried to investigate what could be a possible mechanism for ubiquinone to reach this active site that was at the center of this protein. We reading past literature on E. coli, we found that there's a N-terminal microdomain that E. coli had that was primarily used to associate with this inner membrane. So we conducted a comparison between E. coli DHODH and then the DHODH in the H. pylori that we had, and we used the APBS function actually in PyMol to generate an electrostatic map. Um, by doing this, we saw that there was the same conserved electropositive structure that was primarily associated with arginine and lysine in our N-terminal microdomain. And since we realized that this was also preserved in E. coli DHODH, we began to theorize that the mechanism of association was similar in Helicobacter pylori as well.
okay, great. Anything you want to add, John, to that? What you
Yeah,
said?
sure. Yeah, so I think the significant amount of previous solution phase studies with the E. coli DHODH had provided sort of a standard, I guess, mechanism or model for the association of these bacterial class 2 DHODHs with the inner membrane of the bacterium. And so sort of, with that in mind, and sort of once we found this microdomain and showed that it has this sort of generally positive charge that could allow it to associate with phospholipids in the membrane, we sort of reasoned, okay, well, maybe this, it's not just an association, it's probably, providing some means for the ubiquinone that is known to serve as the electron, I guess, sort of the initial electron donor in these enzymes. We were like, okay, well, is there some channel that could be present in the middle of this? And so we sort of analyzed the structure and found that sort of this N-terminal microdomain, which sort of forms this triangle at the base of the structure of the protein, on its outside has largely these positively charged residues, whereas the inside is almost exclusively hydrophobic residues. And so further exploration of this microdomain, as well as visualization of all of the amino acids within the protein, sort of led us to see that this sort of hydrophobic feature also extends sort of down the middle and connects this microdomain to the active site flavin cofactor. And further analysis in PyMol using the surface feature as well. This didn't make it into the paper, but also visualizing this using the Caver tool, which is an online bioinformatics software, showed that there is in fact this in the case of our crystal structure, what appears to be solvent accessible, but what we would propose is rather membrane lipid accessible upon docking of this enzyme onto the bacterial membrane. And this was sort of one of the, I think, for me at least, this is the most exciting part about this project is sort of seeing the connections between this enzyme that we know functions in a certain way in E. coli and saying, okay, well, based on this structure, we can propose a similar model for that in Helicobacter pylori.
Cool. Okay. So, John, were there any surprises in your search that deviated from what you expected based on the literature or homologs?
Oh, that's a good question. Yeah. So I think, I mean, one of the big, I think, key differences that we noticed in comparing some of these enzymes is that the size of this N-terminal microdomain amongst various class 2 DHODHs does vary. Some organisms add an extra helix that can serve to help bind to the membrane. And so sort of going into this, we knew what the structure of our enzyme was, but from this comparison, sort of I guess, I mean, it wasn't necessarily surprising. I don't think we necessarily had an expectation to begin with, but it turns out that this enzyme sort of has a similar structure to that of E. coli. I think um, probably I don't know if surprising or more maybe
Interesting.
more yeah interesting or
Yes.
rewarding feature was to be able to do this characterization. And I think that's the power of structural biology is to see these molecular features in action that really provide the pathways for chemistry to occur.
Okay. And then you want to add, Ashna, to what John said?
Yeah, I definitely agree. I think one of my biggest challenges throughout this was very early on, we realized that the nature of the static crystal structure itself, and it was definitely intellectually demanding for me to interpret structural biology through a functional lens. So, seeing the previous structures, we initially early on noted that most of the structures that were crystallized and studied, most of the DHODH structures that were studied, had a slightly different conformation because of a bound orotate or a bound substrate, which our protein did not have. So very early on, we noticed small differences in structure because of that bounding. Our protein was obviously crystallized without that particular substrate. So that is something that we had to manage as we conducted this analysis.
Okay, cool. So now I kind of want to switch a little bit and talk about your writing, how you wrote the paper, so the writing publication process. So how did you approach writing the paper? Essentially, how did my class sort of help you guide you through that process? So I'll start with, I think I started with, did I start with Ashna last time? Or I think
Right.
it's John. I'll start with Ashna because I don't remember. But so yeah, how did because I can imagine as someone who's written papers themselves, you're thrown in, here's a structure, write about it, good luck, right? So how did you feel about that, and how did you feel the course helped you in sort of going through that walking with you as you go from a blank screen to a completed paper?
Yeah, I mean, I will start with it really did feel like a blank screen when we started. It was a structure and a blank screen, but I think Professor Smith, your class offered a great framework to start writing. Um, just having the differentiation or having certain goals set out for us. For example, having a goal of separating what kind of analysis we were doing so we could conduct it in a step-by-step manner offered a really good framework for us to have a starting point. And I think even with writing, just recognizing that the first step is simply providing a visual structural analysis of our protein, trying to understand the conserved domains using the cath lab. I think having such a framework really helped us begin. So I think the first few weeks of writing was actually simpler since it mostly relied on structural based analysis using prior literature. I think that was a much more organized framework for us to begin. And I think because of that, we started with a pretty good support system when we then moved on to writing more investigative work, which is trying to look at the domain that we were investigating.
Okay, cool. John, would you like to add what Ashna said?
Sure, yeah. I think one of the biggest sort of aspects of the course that was helpful for us was every, I think, couple weeks or so, there were deadlines for, okay, get the figures ready, right? Know what you're going to talk about, and then write about that with the data analysis and discussion, and then, sort of frame that through the introduction, abstract, et cetera. And I think, sort of having those deadlines was nice for me the first time, writing a scientific publication. I think what was equally helpful was the teaching assistants for the class met with us on a regular basis and gave us feedback on all of our drafts and revisions, as well as other groups of students, other duos working on their own systems. I think that sort of group-based sort of peer review almost at the very entry level of the field. It was great for all of us to see the different sort of layers behind each of these different enzymes, and also see how different people approach solving some of these problems. And I thought it was really useful, not only for writing our own paper, but also just for my own understanding of how people approach science, and maybe I can take somebody else's approach and use it to better my own science.
Yeah, so let's talk a little bit about that revising part, or the sort of revising your work in the feedback process. Yeah, of course, in my course, at least one of the things that I think is really interesting about my course is that during the writing process, it is like a peer review where you work in teams with two other teams, and then you review each other's work, and you provide comments, and then you get comments not only from your peers, then you get comments from your teaching team, right, which is your undergraduate and graduate TAs, and you of course get comments from me. So you get these layered comments, and hopefully those comments, I have a different view of how the project works, and then the TAs will have a different view, and then your peers will have a different view. And so what was it like to revise your work in that sort of process and that feedback? Do you find it helpful? Um, and how did it sort of speak to how you look at science, when you're reviewing other people's work? How did it, sort of, were you inspired by what other students are doing? I know I'm throwing a whole bunch of questions in all this, but I guess I'm asking, how did you feel about this, and the whole the structure and the process, and how did it help you with your paper? I'll go to John with that one.
Yeah, so I think one of the big things was, this for me at least was the first time, doing this sort of peer review type of a thing. And so, I guess at the beginning, I wasn't sure what to expect, sort of went into it, I guess, head first, getting as much as I could out of everybody's comments, but also using it as an opportunity to, I guess, practice my own, I don't, criticism seems like a hard word, but my own, I guess, feedback, how I can provide feedback, I think for others. And I think, going through that process where we each are looking at each other's work and we are being critical, trying to be sure that everybody is representing their work in, the best way possible, that, really does the best job of describing what, nature has given us in these enzymes. And so I thought, for me, it was a really nice opportunity to get feedback, to practice giving feedback to others. I think, having that sort of, layered, I guess, set of people within our small groups, those who are also in the class with us, as well as undergraduates who had previously taken the class, and then, graduate students who have more experience than we did, specifically in the area of structural biology. I think it was very nice, sort of in that small group setting to get that feedback. And then, at the end of the day, when we, sort of had our whole papers together, and Dr. Smith could, give us his feedback, sort of from that, highest, maybe most in-depth level. I think it was, sort of really nice transition to constantly have this revision around the paper, and ensuring that our work was as refined as possible.
Hmm. Cool.
Yep, definitely. I honestly, I truly believe that the peer review process is key to the success of our paper. Truly, when I look back, I realized that all the amount of scientific clarity that we achieved was not possible without the peer review process. A lot of the elements that we've added to our paper came from questions that came up during this peer review process. I think part of the peer review process was us showing up as just the average reader of this paper and questioning each other's decisions, questioning if we've substantiated these decisions, questioning if our solutions actually even answer the questions we're asking. And I think this is such a key part of any scientific paper. And so us having to go through that as a weekly reminder really helped us be extremely accurate with what we're reporting and be intentional with how we're writing about things, making sure that we are interpreting things and we're writing things with clarity and accuracy. I think that was very, very important for us to all to remember. Secondly, I think it just really helped all of us investigate better. I think reading other people's papers also gave us clarity on our own paper, gave us ideas of what are other elements we can incorporate to improve our own writing. Ideas like conserve erupted in our peer review process as an additional way, which we would not have come across unless somebody else had suggested it. And I think it really helped overall our paper and just our ability to write clearly.
Cool. And actually, this particular question is for Ashna, because not only were you students, you came back as a TA. So once you sort of transition from being a student to now you're responsible for responsible for a team, and you have to provide comments. So since you're on sort of both sides of that coin, can you tell me a little bit about that experience?
Yeah, it was incredibly gratifying and kind of jarring at the beginning. I realized in my students when I was a TA, I saw myself. And the first week, everybody came in like, super excited, super confident, and were essentially slightly disappointed when everybody's comments were the exact same, which is this is well written, but it's not scientifically written. This is descriptive writing, except we need to work on formalizing it. I think seeing that really reminded of how John and I came in the first week. We were extremely excited and full of ideas and really needed some refinement, which our TAs were able to help. And at times that was taken with a grain of salt and improved on over time. But I really saw that when I was a TA, that my role was to provide people with support while they're going through this. It is hard to like slowly and deeply work on improving your writing and improving how you're thinking about these questions that you're having. So I think being a student really gave me an ability to provide that support when I became a TA as well. And yeah, just watching people like slowly get more confident with working with these tools and definitely develop a stronger acumen for writing about it was incredibly gratifying.
Yeah. So yeah, and that's actually my favorite part of the course actually is watching development, right? So watching that students really start with nothing and then that final check out day, right? When I'm checking you guys out, making sure you have everything there before that last day of class and to see the growth. And because I retain all the first draft and all the drafts. I see all the drafts. So I see that growth and development. And so it's really nice and gratifying to see. And hopefully you guys can sort of appreciate that, the growth that you've done throughout the entire semester. So, John, what's some of the important skills that you gained out of the course that you sort of can carry on into your sort of future professional plans?
Yeah, so I guess I'll highlight two, maybe three things. So I think the first thing is, coming into this new fresh project, I hadn't, I knew nothing about Helicobacter pylori, dihydroorotate, dehydrogenase when I started. But by the end of it, I think one of the biggest things that was helpful was you just learning how to approach the literature in a new field that you haven't studied before. I think it was a very, I guess, formative experience for me, approaching specifically, a lot of structural biology literature to try and understand how we can frame our own paper and how that adds to the conversation there. But also some of the studies with this particular enzyme and flavin enzymes in general. I think it was a really useful experience to enter into a new area that I hadn't been in before, and I learned a lot, I think, from that process. I think the second thing is probably equally as helpful now that I'm doing further research in enzymology is how do you approach looking at the structure of a protein. I had done a little bit of structural visualization previously, but this class was really the first opportunity I had to work with PyMol and a number of other different softwares that you can use to visualize proteins, that you can use to analyze their structures, as well as make structural comparisons between these. So it was sort of my first foray into that structural bioinformatics area. And it's been incredibly helpful so far in trying to look at crystal structures of some enzymes, but also, structures generated by AI with tools like AlphaFold and bolts that have really sort of revolutionized how we're looking at structural biology and enzymology as well. So I think that's sort of the second area that's been very helpful for me. And third, I think was just it's I guess it's maybe the softest skill of all, and that's just a general excitement for not just science, but specifically for enzyme science, understanding how these enzymes work, understanding how structure turns into function, and how that can be evolved to maybe better the catalytic capability of these enzymes for use maybe in industrial applications, or how we can understand enzymes at a much deeper level than we did before, and how that can maybe inspire the development of new catalysts down the road. Maybe that synthetic chemists could use. So I think maybe this sort of third thing that I took away from the whole process was just I mean, science is cool, enzymes are cool, nature is fascinating, and that was really exciting to sort of come to throughout this process.
Cool. All right, Ashna. So, what would you like to add to that?
I mean, I definitely agree with John. Like a lot of this similar implications, I think for me. Yeah, I think I really, as a student and then even as a TA, I realized that the structural based approach that we were like learning was it goes beyond just a single protein. For me, it went beyond just Helicobacter pylori. I realized that the structural characterization of these enzymes could really have a large impact. It could really inform the development of new target treatments in our scenarios, but then also could really be a key pillar of working on advancing collaboration across different fields. We really worked with the work of structural biologists, chemists, etc. I think being in this class really taught me, it just inspired me to work towards researching novel ways. Like I think learning these computational tools really helped progress my scientific inquiry and my interests itself. Um, and yeah, I think just learning how small tools like this in collaboration with people and people's work and making it accessible can create such a difference really pushed me to work towards uh, not only scientific discovery, but making it more accessible to people.
Yeah. So, well, yeah, so then on that note, has this influenced your career path or what your future plans are going to be, or or changed the way you thought about research? Did you have an opinion what research was before? I think both of you guys have had experience doing undergraduate research in other capacities, but so after taking this course, did it change how you thought about research or augment or solidify what you already thought? So what would so how did, yeah, what do you guys think about that? Anyone could jump in and then have the other comment.
Yeah, I thought honestly, I think it expanded the way I thought about research. In my prior experience with research, I worked more with more rigid organization. I worked with more experimental tools, which are much more rigid, much less accessible. And I think having the opportunity to work with a structure-based, software-based design, I think it taught me completely novel way of understanding science. I think realizing how data rich and shareable this methodology was really expanded my idea of research. I think in our project, we were able to not only foster collaboration across structural biologists, chemists, pharmacology, etc., but also I think like our research was a conclusion of it. It was a way to have a common visual interactive language that multiple people can now work on. I think the work that we were able to publish is now a blueprint for other labs basically to explore and further study or design inhibitors for. I think having that be so accessible was beautiful, and I think that gave me a new perspective.
Hmm.
Yeah, for me, around the time that I was in this class, I was really starting to question what I thought I wanted to do for the rest of my life. I came into college firmly as a pre-med, and around this time, I was, sort of thinking, okay, maybe I want to do, not just an MD, maybe I also want to do a PhD. And really, by the end of this class, I decided that I just wanted to do a PhD and fully dive in on the research end. So this class was, I think, really transformative for that. It was, I'd done some other research previously, but this was a really great opportunity for me to jump in, take hold of a project, develop it, grow my own understanding in this area, and then write a paper from it. And I think having that experience really sort of showed me what it might look like to eventually do a PhD, for example.
Mm,
And so I think the big takeaway that I got from this class was experience and maybe a drive to learn more and to continue to do research.
cool. And so what advice would you give students considering taking structural bioinformatics or proteins and or any other advanced labs? Actually, let's concentrate on Bio 4525. So this structural bioinformatics class that you took, what advice would you give students considering this course?
Yeah, I guess I can start on this one. So I think the biggest advice I can give is that, from the day that you start the class, and the day that you, find out what your structure is, read as much as you can. Not just about your enzyme, but maybe about, where that enzyme falls in some larger metabolic pathway. Understand the context, the why behind why we're studying this enzyme in particular, why it might be of interest for drug development, for example, or why it's something a mechanism in the cell that's used for some important process and why we care about that. I think having that sort of motivation that you get from understanding, why this enzyme is important, I think can help you, during the times where, studying your system, you don't really know what's happening, but you can always come back to that North Star that you know why you're doing this. And that, the whole process is a learning experience. You're meant to make mistakes and learn from them. So trying to fail, I guess, with grace, and get back up again and try again and maybe fail again, but that's fine, right? Because we have this safety net of people around us that are trying to help us become better scientists as we try to help them as well.
Cool. Ashna?
Definitely. I think my biggest advice would be to come in open-minded. Be open-minded to what you're seeing in front of you, to the protein you have, to all the criticism you're going to get, because it is incredibly useful. And really be open-minded to what you see in your protein. There's no finding or uh, fixed conclusion that you are to come to. There's no fixed process for this kind of class. I think the best part of this class is being able to independently work out the process of where you're going to go with the protein, investigate using all of the information that you have, including what you see in front of you, what the path should be. I think for us, what really helped was realizing that it wasn't just about observing domains or just observing these active sites that we had seen. It was really about connecting those features to the biochemical activity of this protein. And I think just realizing that was super helpful, and I think would help a lot of students.
Cool. So actually, I guess one last question here. I'm just really interested in hear what you guys are doing now. So, Ashna, what are you doing now? And then, and what you're planning on doing in the next several months, or what's your sort of career plans? And then I'd like to hear what's going on with John.
Yeah, currently I'm working as a patient care technician at an urgent care in St. Louis. So I work with patients every day. Usually it's Um, usually it's a mix of expert, like professional and non-professional services, but usually I am either doing that or I'm volunteering with CASA, which is a organization that works to improve the lives of children in the foster care system in the US. So that's what I'm currently doing. My future plan is to apply to medical school hopefully soon.
Hmm. So, are you applying this cycle now, or are you applying? You're gonna wait another year?
I'm currently in the cycle right now,
Okay,
but
cool.
I will hear back by next year, I think.
Okay, great. So, I'm sure you're gonna be in some medical school this time next year, right? You're probably going to have a choice of your medical schools. I don't think that'll be a problem for you.
Hopefully.
All right, John.
Yeah, so after graduating from WashU, I guess last spring, I started my PhD in chemistry at Princeton University this past fall, where I'm now working in Mohammed Said Sayamdost's lab, specifically focusing on characterizing the mechanism of radical SAM enzymes involved in the biological synthesis of RIP natural products. These are peptide natural products. We're specifically interested in those that form microcycles installed by these radical SAM enzymes. I'm also, interested in other natural product biosynthesis and characterizing the mechanisms of other enzymes. And I'm really excited to see where this goes. I think long term, I want to stay definitely, in this enzyme space. I guess, I've committed 5 years at least to working on a PhD in this area. Very long term, I think it would be really cool to run my own research lab and to teach in a university. But there's a long road ahead, and I'm, going to keep my head down and keep working now.
Mhm. All right, great. It was wonderful talking to you guys. It was wonderful hearing, how you felt about my course and how you felt about the whole paper writing process. I just want to add, you guys did a fantastic job on your analysis, and I think I had told you that, during the peer writing process, the reviews I got back from the paper, really, unanimously said that this is a well-written paper, and so your hard work really showed up in the reviews. I was very proud to hear the reviews that your paper is well received, and it was sort of evident all the hard work that you guys did. So I'm very proud of both of you. And I wish you both, good luck in your future endeavors. Both of you are going to be doctors, it sounds like, at different types, a PhD doctor and an MD doctor. And I'm looking forward to hear all the cool stories and the cool things you're going to be doing. So you guys take care.